the index would be 0, but it basically tries to quantify the extent of help that you get from one organism to the other. And we've also extended this to communities. So that was the source of one of the questions I had asked on Slido. Because sometimes you have, there could be some confounding from very similar organisms that are present in the medium. Right? So if you have two strains, similar related strains of E. coli, they could be contributing very similarly to the community, and you really can't disentangle that to understand what is the impact of one organism on the rest of the community. And we studied this in many cultures. So one of these is we did the experiments ourselves, trying to study Pichia stipitis and Saccharomyces cerevisiae. But we also looked at a bunch of other cultures that people have studied. And wherever we found that one organism was getting more help, it seemed to have a higher biomass. So in all of these cases, the organism with higher metabolic support had a higher biomass in the co-culture. So this was only pairwise interactions. I have not gone into higher order interactions yet. But essentially, our graph models help address many questions. And this is something I want to emphasize for all of you. What are the metabolic capabilities of a single organism or a community? What kind of metabolites could possibly be exchanged between these organisms? How do these organisms depend on each other and survive? And what are the higher order dependencies? For example, in a community, or do you have unique contributors of particular metabolites which give them a more keystone sort of role in a community, and so on? Very nice study. I think Aarti explained most of it yesterday, so I can breeze through this and focus on the other studies. But this is a very nice representation cartoon of what happens to the gut when you take antibiotics. It's like a forest fire where the entire gut is laid waste. But then, the ecosystem slowly recovers. I think we saw a few better pictures, even from Alex, about how they could recover mangrove ecosystems by probiotics and so on. But essentially, what is the probiotic concoction? Here in this study, the question was, are there individuals who recover faster? And they seem to have certain organisms in their gut. Can you use those? Can you understand what makes them recover faster? So they identified about 21 recovery-associated bacteria. And we looked at very specific interactions and synergies between these organisms, again, by doing pairwise simulations. But it would be very interesting to look at higher order simulations as well. But we basically found that the recovery-associated bacteria were working together very much as a community. They were really benefiting from each other. And we also looked at a few metabolic and so on. And this particular was one of the strong edges that we predicted. Theta, iota, omicron was really helping bifidobacter placentis. And this was evaluated in a mouse model. It seemed that, there was a very fast recovery of microbial diversity when this was given to, when this combination was fed as a probiotic to mice. And we've also looked at different interactions in the gut microbiome. We've been looking at trying to understand what kind of organisms play a key role in the gut in general. Many interesting questions. So how do they interact? How are they shaped by diet? And are there particular groups of organisms which behave differently, and so on? And we looked at 52 commonly occurring organisms in the gut and pairwise models and studied them in two representative media. There is a minimal gut medium and a high fiber medium. We again looked at metabolic exchanges and support. I'll just quickly cut to the chase. So we found that fusobacteria derive a lot of benefit from other organisms. And we find that some of these interactions obviously change when you change the medium, but others are fairly conserved. And there are many interesting patterns that we are observing, and we are connecting these to what is observed in literature. But we obviously want to see if there are other experiments that can be done to understand these more deeply. We find that in a high fiber diet, the organisms are more independent, a kind of environment, which is expected. But even so, there are still some strong dependencies you find in those environments. So this basically underlines the fact that there are some very strong dependencies, and these organisms cannot just function very independently of each other. And this is a network we built interactions, and we find some species that are very highly connected and so on, which have very interesting metabolic signatures. I'll not go into all of the details now. And we also looked at how there are some lactic acid bacteria which enrich the growth of other organisms and of amino acids between them, and so on. So essentially, you can explore a lot of these exciting questions, and also what organisms produce butyrate, propionate, and so on. And we focus on these because we already know that these short fatty acids have a big role to play in the gut to mediate reactions and so on. With that, I'll quickly touch upon this nice which we did with Venkat, and I think Venkat has already briefed you on some of this. This was our approach. We focused on the locations with where Klebsiella pneumoniae was present, and then looked at the organisms which it coexisted with. We looked at both a network-based analysis using MetQuest and also a constraint-based analysis. So I think this multi-pronged approach is very helpful. And here we did both a pairwise analysis and a community-based analysis. This community-based analysis essentially leaves out a set of organisms, not just a single organism, like an entire, all of, like Pantoya or something like that, and then tries to see what is the impact on the community. This helps quantify what is, sheds more light on what kind of interactions can exist with these organisms. And we found that Pantoya species were highly dependent on other species for survival, especially Klebsiella. And we also predicted a bunch of amensalistic and parasitic interactions. And Venkat validated one of those predictions where we looked at the interactions between Klebsiella and Aspergillus. So to summarize, we believe that these graph theoretic approaches and metabolic support index are very helpful approaches to study microbial interactions. And it's important to look at complementary approaches, right? So you have each of these approaches give you a different lens to into the cell, and it gives you a more complete picture when you use multiple approaches. And we felt that these pairwise interactions and community interactions are really helpful. We did leave one out and leave an entire genus out. Studies to understand what is the role of a single microbe or a family in these kinds of communities. But as Venkat was planning yesterday, there are so many more predictions we make than we test. And I think we really need to understand a little bit more, but it provides us very interesting insights for clean room microbiomes and how to disinfect them and so on. The last bit of study is ongoing. We have a lot of interesting things here, and but I'll just quickly walk over this. I don't think I have to explain hydrothermal vents because we saw some pictures and descriptions already. But basically, there are very unique microbes with very unique metabolisms that you find there. And we again find very strong dependencies between only a handful of microbes. And we are now trying to use these to see if we can culture these microbes, which are typically otherwise uncultivable in the lab. And so can you grow these in the presence of each other because there is such strong dependencies? And we find that the signatures, so these are the resource overlap between the hydrothermal vent kind of scenarios and gut scenarios are starkly different. So we find that there is a very interesting kind of metabolic overlap between these kinds of organisms. So essentially, there are so many interesting studies that we can perform using the metabolic knowledge of these organisms. So I'm reaching the end of my talk. So to summarize, there are some limitations that I wanted to anyway point out. Most of our graph models provide a static snapshot of the interactions. Of course, as if you have time course data, you can look at how these interactions change. But in essence, it's a somewhat static picture. And as I said, many predictions remain to be validated. And of course, we are very dependent on the kind of data that we have, the quality of the input network that has a big effect on how we build these models. And we've not yet been able to attach weights or rankings to these paths. I think this is something that we want to really do soon because it's to identify what is the most important contribution from one organism to other, whether it's a serious, substantial contribution in terms of metabolism. And of course, I've been saying only metabolism from the time I began the talk. There's a lot more happening in reality, and that's something we need to be cognizant of. And I think I've said all of these. Just leave it at this slide. Just to tell you, though, but there is a generic framework to understand microbial interactions, and you can really use this on any system that you're interested in. And we've been able to see how the community structure is driven by some of these interactions. So that's really interesting. There are many open questions that we are very interested in. So if this interests you from any other perspective or any other system that you're working on, we'd love to have a chat. What are the key differences between pairs of microbiomes? Could be healthy versus disease, rural versus urban, for which we are building tools for a differential analysis of these networks. And something about the dynamics, how do these assemble? Right? So are some of these very resistant to invasion, some certain microbiomes? Can they be easily colonized by another invading species and so on? Or can you manipulate them by using prebiotics or probiotics? How can you influence the trajectory of these microbiomes? And we're looking at minimal microbiomes that may also be helpful to study some of these systems in the lab. So these are some of the things we are currently up to. So with that, I'd like to acknowledge host of my students and collaborators and our funders. And I think I'd really like to especially acknowledge Aarti and Rachita and of course, Venkat. So with that, I'm ready to take questions. Thanks for your attention.
Oh, a very interesting thing. Something occurred to me that you are extracting the shotgun from the metagenome. You are predicting all these metabolic, genes responsible for different metabolites. But we have to think about few. One is when you are assembling these genes, 80% of them we are not actually considering, we are throwing out.
Yeah.
That's one. Second one is when you are extracting DNA, there is no one method extract every single thing. Okay. And that is actually allowing these dominant microbes to grow. Okay. I'm not asking you to answer this question. So these are some of the suggestions. The third important thing is the rare microbiome, which is tail, because 3 or 4 or 5 communities, constitute 90+%. We don't know the rare microbiome. Because of that, we don't have the genome. They are not in culture. We don't have the genome. We don't know what it does. Maybe your students and others, the next generation scientists have to think about this first, how to get that 80+% that we are throwing away into some meaningful genes and other things.
Absolutely. And I think that's exactly what I was talking to Alex the other day, right? Because these are such novel microbes, so very little help you can get from existing databases and so on. So I think we have to, maybe with the level of data we have, we can actually build some machine learning based methods for this. I think that's what we are dabbling with right now. We have to see how successful these methods are. But I think there seems to be so much data that we could actually go in for a machine learning based approach here. But yes, I think these are very exciting questions too. And clearly, for example, I think the tail doesn't matter, if their abundance is not high, but they could still be producing so many unique metabolites. Or maybe, they are producing some bacteriocins or some antifungal compounds that are really upsetting the entire community, right? Even though they are in small concentrations. So I think this is something that we will get closely. Thanks, Venkat.
Sir, it's a nice talk. Like, I just want to know about a bit about this algorithm. Like, I'm completely new to this microbiome field. Like, in comparison to pie crust, how you will explain like how it's different from the pie crust, this algorithm?
So pie crust is an assembly sort of pipe, right? Like, so this looks at given a single microbe, it tries to characterize its metabolism. And when you put it with another microbe, how would it interact? So I can show you a little bit more, a few more slides that I can explain to you. Maybe we can catch up over coffee.
Thank you, sir.
Hi, sir. Very nice presentation. I wanted to ask, like, when you're talking about the environmental microbiomes, say, for example, Arctic, where I have worked. So there are so many environmental parameters also involved in the interactions between the communities and also their metabolism. So how do you incorporate those environmental factors? Like, do you have...
Excellent. I think, for us, finally, this becomes like a bag of reactions. Right? So if you have some transcriptome or any other study or any other environmental related measurements that can predict what reactions are going to be active and inactive, I could totally use that and we would get far better predictions. So basically every microbe for me is a bag of reactions. So what are the reactions that go into that bag? We have to sort of estimate from, say, other data, could be transcriptomic or whatever.
Thanks.
Yeah. So can graph-based approach be to predict temporal evolution? I'm going to point you to Gayatri in the crowd there. She'll answer it for you. And what type of are upregulated? I'm not so sure. So typically we find a lot of metabolites like pyruvate, acetate, and so on being exchanged. And depending upon the organism, a variety of metabolites are actually produced. If you look at these, the guaimas, very exotic microbes, very interesting sulfur metabolisms, and you see that whole pathways are actually enabled by the presence of another organism. And can we use the same approach to predict metabolic interaction between hosts and microbes? Yes, people have done it. Lots more challenges because the host metabolic models are far less accurate than what we would like them to be at this point of time. But there are very interesting approaches. I can point you to some of these.
Thank you, Karthik. And on that note, let's thank Karthik once again for a fantastic presentation.
Thank you.
Let's move on to the next talk today.